Can we use COVID to fight COVID? Researchers think that's possible. Novel drugs, repurposing medications, and others are being tried to end the ongoing pandemic. But what if we can use COVID 19 against itself? This is the angle under scrutiny by researchers at Penn State. They designed a proof of concept of using the novel coronavirus to eliminate SARS-CoV 2. While it sounds weird, they demonstrated in lab settings how this concept may lead to potential therapy. Be guided, however, that the study is still at the infancy stages. When the coronavirus enters the human body, it looks for cells that have the ACE2 receptor. Unfortunately, many human cells have this receptor on their surfaces. To prevent the virus from attaching, something must block its ability to connect to host cells. So, scientists created vaccines and antibody therapies to hinder both the connection and replication of the virus. In the new study published in PeerJ, Penn State researchers used a defective SARS-CoV-2 based on the approach called defective interfering viruses. These viruses are common in nature and contain deletions in their genomes. These deletions negatively affect the replication process, making copying the genetic material and turning it into virions difficult. But there's a way to exploit this. Like other viruses, DI viruses must have host cells infected with a wild virus. During an encounter, DI attacks the infected cells and hijacks the normal machinery of the wild virus. It's like DI can interfere with the wild virus by inserting its defects into the machinery to make its own copies. These defective genomes are like parasites of the wild-type virus, said Associate Professor of Biology Marco Archetti. Researchers engineered short synthetic DI genomes from parts found in wild SARS-CoV-2. Next, they introduced these genomes into African green monkey cells, which have been infected with the wild coronavirus. Then, they quantified the relative amounts of the DI and wild-type genomes so they would know the level of interference. Results showed that in just 24 hours, the DI genome decreased the amount of SARS-CoV-2 by about 50% compared to the wild-type in control samples. Plus, the DI genome ballooned its numbers 3.3 times faster than the wild coronavirus. Although the findings are promising, the 50% decrease in the number of wild type isn't enough for therapeutic purposes. The DI genome may just wipe out the wild SARS-CoV-2 given the 3.3 times faster replication. It would lead to the extinction of both viruses. Thus, more experiments are needed to confirm if the DI genome can result in an antiviral. If yes, experiments involving human lung cell lines and COVID variants would take place.